Cell membrane is also known as plasma membrane and lipid bilayer. It is mainly composed of phospholipid and associated protein. Phospholipids is made up of two parts, one a phosphate head, which is polar water-soluble hydrophilic and compound having charge, and a tail is made up of fatty acids, which are non-polar hydrophobic. Fatty acids dissolve in each other and form the lipid bilayer, two layers. Lipid soluble and very small molecular weight compounds can pass easily through the cell membrane, but the charged and large molecular weight compounds will be repelled, making it semipermeable membrane. Lipid soluble substances such as oxygen, carbon dioxide, and steroid hormones cross the cell because they can easily dissolve into the hydrophobic lipid bilayer. However, water soluble substances cannot dissolve the lipid membrane but may cross through the water filled channels or pores or may be transported by the carriers. Intracellular environment is aqueous environment and extracellular environment is also aqueous environment. So the phosphate heads press towards the aqueous environment and the fatty acids, which are non-polar, dissolve into each other. The outer leaflet of cell membrane is asymmetrical to the inner leaflet of the cell membrane. Due to different phospholipids, different proteins and carbohydrates are only present on the outer leaflet. The phospholipids present on the outer leaflet passing towards the extracellular environment are phosphatidylcholine, sphingomyelin, and phosphatidylethanolamine and phosphatidylserine pass towards the intracellular. One which is present in the cell membrane is the cholesterol, which is present between the fatty acids. Cholesterol trades with the hydroxyl group passing towards the aqueous environment. And the other thing which is present is straight structured are saturated fatty acids. The structures which have that change in the structure are unsaturated fatty acids. Lipids are mobile and move laterally. This is known as the property of fluidity, and which helps in the secretion of hormones by the process of exocytosis. For example, secretion of insulin from the beta cells of pancreas, and the other process of endocytosis. For example, for the WBCs, when the endocytose pathogen, they kill the pathogen by the process of endocytosis. And the other important process, mitosis and meiosis, which is membrane biogenesis. This fluidity of the membrane allows to the membrane that change the form. This change in the form causes these secretions of hormones, means exocytosis, and phagocytosis, endocytosis, and the membrane biogenesis. Fluidity depends on the following factors: the increase in temperature, increase in fluidity as the movement of molecules increases. Increase in cholesterol decreases the fluidity as it tightly packs the membrane. Unsaturated fatty acids, if they are increased, fluidity is also increased. They are directly proportional as they have different structure, so it creates the space. Saturated fatty acids decrease the fluidity as they causes the packed cell membrane. Receptors are macromolecules, usually proteins, to which when a specific substance, neurotransmitter, drug, hormone binds. Receptor gives signal to the biological system and alters the function of the biological system. If the stimulus is highly polar and large, then the receptor for that substance is present on the cell membrane, like this one. But if the stimulus is lipid soluble, small, non-polar, the receptor for this substance is present inside the cell. Signaling mechanism is like lipid rafts. Receptors and their associated proteins are rafting on a cell membrane in specialized area. Specialized area, that means phospholipids are tightly packed, fatty acids have long chains, and
cholesterol is present between them. They are tightly packed. It is a specialized area where the receptors are present. There are two types of receptors: integral proteins or transmembranous proteins, those proteins which pass through and through the cell membrane, like these green ones. They have extracellular domain and an intracellular domain. And others are peripheral proteins which are loosely attached to the membrane, like this pink one. They may be attached to the external or internal side of the cell membrane. These proteins are attached in non-covalent ways that they can be easily removed from the membrane by slight change in pH or ionic composition. So the proteins make up to 50% of the cell membrane, and the lipids make 47% of the cell membrane, which mainly phospholipids with cholesterol and other lipids. Carbohydrates make 3% of the cell membrane. Proteins may work as a channels. Channels are made up of peptide chains. The central area is filled with water. This is the aqueous tunnel through which the water-soluble substance can pass easily through the membrane. Carrier proteins, when they bind to a specific substance, they undergo transformation. They change their shape and transfer the substance across the membrane. Enzymes take one substance and convert it into another substance, like this one. A plus B is a substance uh, to this protein, then this protein changes it into C plus T. This is the enzyme. Integral protein work as an enzyme too. Linker proteins bind the internal cytoskeleton with extracellular compound. This example actin is present inside the cell. This is the cytoskeleton of the cell. Skeleton of the cell. Just as we have the skeleton like bones. It attaches to the outside extracellular component collagen. It helps in the stability of the cell. Other integral proteins work as receptors. Receptor has extracellular, transmembranous, and intracellular component. Substance bind to the extracellular component, and intracellular component produce the signals. For example, if insulin binds to the extracellular uh, component, intracellular component transfers the signals, and the uh, transfers the signals, produces signals, and there is a uptake of glucose.
Proteins, they can be attached internally or externally to the cell membrane. They are loosely, non-covalently attached. They are held with very weak electrostatic forces that they can be easily disrupted away from the cell membrane. Number one, they act sometimes as a component of cytoskeleton inside the cell, which may be attached to the linker protein. This one, for example, is a linker protein, and this sometimes act as a component of cytoskeleton. These are the peripheral proteins present inside the cell. Number two, receptor proteins like integral protein like this one are sometimes associated with the peripheral proteins. Peripheral proteins which are associated with second messenger system are peripheral proteins, not just the integral proteins, but the receptor is the integral protein. First messenger, for example, hormone, uh, binds to the receptor, which is integral protein, and this receptor, which is peripheral protein, produces the signals. This works as a and stimulates the second messenger system.